In this video, we will be identifying the localization of SARS-CoV-2 virus using its genome and tools such as the BLAST search and Snapgene to insert a gene of interest in a plasmid, then into an animal to identify its localization patterns. The agenda includes a brief introduction about COVID-19 disease and SARS-CoV-2 as the causative agent. Then we will utilize BLAST search and Snapgene to find and insert our gene of interest into a bacterial plasmid. Transformation, selection, and detection will also be covered. The significance behind studying this topic is that the 2019 novel coronavirus, SARS-CoV-2 virus causing the disease COVID-19, was identified as the cause of a cluster of pneumonia cases in Wuhan, a city in the Hubei province of China. It rapidly spread, resulting in an epidemic throughout China, followed by a global pandemic. The COVID-19 infection originated in bats as the primary source of infection. Pangolins were the intermediate carriers through interspecies transmission, followed by the infection to humans. Airborne transmission is the main form of communicating the infection, though surface transmission occurs as well. And the R node, denoted for the pathogen's basic reproductive number, represents the average number of people infected by one infectious individual. If R node is larger than 1, the number of infected people will likely increase exponentially, and an epidemic would arise. If R node is less than 1, the outbreak is likely to peter out on its own. The R node for SARS-CoV-2 infection is 2.5, approximately 3, which means that every infected individual is likely to infect 3 more individuals. Severe acute respiratory syndrome coronavirus SARS-CoV-2 is an enveloped, positive-sense, single-stranded RNA virus with a genome of approximately 30 kilobytes that causes coronavirus disease, the COVID-19. The structural proteins of SARS-CoV-2 include the envelope protein, spike or surface glycoprotein, membrane protein, and the nucleocapsid protein. The spike glycoprotein is found on the outside of the viral particle and gives coronavirus their crown-like appearance. Early in the infection, SARS-CoV-2 targets cells such as the nasal and the bronchial epithelial cells and pneumocytes through the viral structural protein S protein that binds to the angiotensin-converting enzyme ACE2 receptor. The most common signs and symptoms encountered with the COVID-19 disease would be fever, dry cough, shortness of breath, while the most common complications include pneumonia and acute respiratory distress syndrome. Pathophysiology. The cycle of infection begins when an infected person talks, coughs, or sneezes. As you can see in the video, the droplets containing the virus will be transferred to another individual, where it can attack cells in the nose, throat, or lungs. The virus will be attached to its receptor, the ACE2 receptor, via its spike proteins, which will allow the internalization of the virus inside of the cell. The virus doesn't even have to go inside the nucleus. Instead, it employs the ribosomes to create more and more of its components, such as the spike proteins and others, which will then be transferred via vesicles to the outside membrane, where they will form together and bud off as one complete viral entity. The significance of our work is that we will localize the viral infection to identify the specific parts of the body where the infection is limited to due to the abundance of its target receptor, ACE2 receptors. This helps us understand the patterns of the symptoms and why some systems are more affected than others. Once the virus is localized, vaccines are to be considered after. The basic local alignment search tool BLAST finds regions of local similarity between the sequences. The program compares nucleotides or protein sequences to sequence databases and calculates the statistical significance of matches. BLAST can be used to infer functional and evolutionary relationships between sequences, as well as help identify members of gene families. We begin by Googling BLAST search. And then we click the first result in the Google, and we go to the nucleotide BLAST, where we will insert our full genome sequence of the virus. We go to the first link and click on the accession link to view it. We will find all the different components of the virus with the different proteins. Our target here is to find the sequence of the S protein, since it's the one responsible for the attachment and internalization. You can see this is a glycoprotein, but it's not the spike. This is the spike protein. And the sequence here is given as a protein sequence, not a nucleotide sequence. This is why we will be copying this sequence, going again to the BLAST search, this time using the T-blastin in order to convert our protein sequence into a nucleotide sequence. We click BLAST, and then we will have the different results. The second result is what we're looking for because it provides all the different types of proteins separately. It doesn't show an accession link, so we click on the traditional view, and now we can view it by clicking on the accession link. Again, we hunt down our spike protein, as you can see. We click on the CDS to take us to the nucleotides. Then we go to the gene bank just to make sure that we got our spike protein alone. Yes. So now we can actually save our file as a coding sequence, and this sequence is what we will be using in the Snapgene. Snapgene is molecular biology software that allows users to plan, visualize, and document molecular biology procedures. For cloning and expression, we use the DNA sequence of the SARS-CoV-2 spike protein. We can see that we have inserted restriction sites, the SAG11 and the ACC 651 before the start codon and after the stop codon. We also added a green fluorescence protein at the beginning of the start codon. We clone our gene of interest into a pBlue script vector. It's a shuffle vector that clones and expresses at the same time, and the reason why we used a positive sense vector is to be compatible with our positive sense RNA genome. In the multiple cloning site, we can see that we have a bunch of restriction enzymes, two of which are the ones that we have used, the ACC 651 and the SAG11. On the right is our gene of interest with the two restriction enzymes. On the left is the plasmid of interest with the same two restriction enzymes, both of which have to be in the same direction for the correct insertion of our gene. And this is the final plasmid, where the red band is our gene of interest. As we zoom in, we can see that our gene of interest was inserted correctly into the multiple cloning site with the GFP, green fluorescence protein. The LAG-Z in this case was not disturbed, and the ampicillin-resistant gene is available in the plasmid, and this will help us later on with the selection. And here we have the final recombinant DNA ready to be inserted into the bacteria. After obtaining the plasmid with our gene of interest, we must insert it into the bacteria, a process known as transformation. There are two types of transformation, the chemical transformation and electroporation. In the chemical transformation, the bacteria is subject to a heat shock to create pores in the bacterial cell membrane to facilitate the entrance of our recombinant plasmid. A similar concept is used in electroporation, but instead of using a heat shock, we employ an electric shock to disturb the membrane. For plasmid selection, there are two methods: antibiotic selection plate or blue and white selection. It is important to note that the plasmid with our gene of interest contains ampicillin resistance gene. Therefore, when we transfer the bacteria from the control plate on the left to the ampicillin-containing plate on the right, only colonies with plasmids containing the gene of interest will survive. Another method of selection is blue and white selection. This happens when we disturb the LAG-Z gene upon inserting our gene of interest. So the product, beta-galactosidase of the LAG-Z gene, will not be produced. We transfer the colonies to a plate containing ampicillin and X-gal, and only colonies whose plasmid carry a functional lag Z in the polylinker can metabolize the X-gal in the media, resulting in blue colonies, while those with a polylinker disrupted by an insert have a non-functional lag Z gene and are unable to metabolize the X-gal. Therefore, they produce white colonies. Detection and localization of the GFP-linked spike protein. An injection containing the E. coli with the cloned plasmid is injected into a mouse, where the mouse is killed by cervical dislocation and various organs are scissored out. Part of each organ is pinched out and pressed under a cover glass before detection. Either perfluoroalkoxyalkane or methanol is added for fixation, and then fluorescence microscope emitting a specific wavelength is used to detect the GFP fluorescence to determine the viral localization patterns.